Beanbag genetics, Wikipedia article audio. Beanbag genetics is a conceptual model of genetics which was used by early Mendelians, who used to keep colored beans in bags as a way of tracking Mendelian ratios. To be able to understand beanbag genetics, the meaning of population has to change. Population is no longer a group of individuals in an area but are all the alleles in an area that assort and segregate separately. All of the alleles become the gene pool. When using the beanbag approach, there are two ways that the gene pool can be viewed. The first is to view the gene pool as all the alleles that represent all the traits in the population be viewed at once. The second, the gene pool could only be alleles for a single trait in the population. Once the alleles are chosen for the gene pool, they are selected for at random. Phrase was first coined by Ernst Mayr in criticizing the work of Ronald Fisher and J.B.S. Haldane, who treated genes as independent entities to simplify their mathematical analysis of population genetics. Mayr created the name, beanbag, because all the alleles were thought of like beans in a beanbag. The beanbag full of beans would be considered the gene pool for the population. The paper Perils of Industrial Gene and Beanbag Genetics, written in 2008 by Drive-Lock OSHA, discusses that biotechnology needs to be reassessed before continuing down the path that it is going down now. Dr. Lock OSHA believes that overall everything in our genome needs to be looked at rather than just coding regions. We do know that our genetic material forms an organic system that is built by multiple smaller units working together. Since we know that multiple smaller units are used, scientists need to look at more than just the structural gene. We should look at the entire genome because know that even in the non-coding regions of our DNA is involved in the regulatory network. Studies. Debate. The debate of beanbag genetics has been occurring for many years now. One thing scientists have agreed on is that beanbag genetics is too simplistic and needs to be built upon. Sir Ronald Fisher and Sewell Wright are two scientists that were once a part of this debate. Fisher's assumptions were that traits are determined by many loci, populations are well mixed, and mating is random. Wright's assumptions were his shifting balance theory that included random drift, individual selection, and interteam selection. As both argue their theories, people agree with Fisher's theory because it works and is a well-developed theory. Other people agree with Wright because his theory is closer to the real world and provides an aesthetically pleasing view of evolution. Even though both theories have parts that are right, they both are wrong as well. Fisher's theory does not explain the diversity of the world, and Wright's theory is just a metaphor, not a well-developed theory. So how do they know which theory is correct? Even though both theories are right and wrong, they both see a part of a larger picture. Citations.